We are live, sir.
Yeah, please.
Good afternoon, everyone. I, P. Kitisika, working as assistant professor in the Department of Pharmaceutical Analysis, extend my hearty welcome to the respected dignitaries, honorable speaker, distinguished guests, organizing committee members, my faculty, and dear students to this Indo-African International Webinar Series, Part 1 on the topic Role of Pharmaceutical Analysis and Quality Assurance in the Vaccine Development, organized by the Department of Pharmaceutical Analysis, Sri Padmavati School of Pharmacy, Tirupati. Today's theme is very apt to the current situation where each and every pharmaceutical company is focusing on the vaccine development and what is the role of the analyst and pharmaceutical analysis in developing the vaccine is the major theme of this today's topic. I am overwhelmed to say about the registrations what we got received for this today's session. Nearly more than 500 registrations from the students we received, not only from India, not only from India, we also received the registrations from various countries like Kenya, Malaysia, and other African countries. So I wholeheartedly thank all the students for the active participation in this today's session. Today's session is going to be really very useful and informative. Now, I would like to invite all the dignitaries and the organizing committee members to the today's program. First, I would like to invite the coordinator of today's Indo-African International Web Series, Dr. A. Srinivas Charan, sir, to the program.
Thank you, one and all. I welcome to international webinar. Thank you.
It's my pleasure to welcome the organizing secretary of the today's program, Dr. K.K. Rashekar, sir, head of the Department of Pharmaceutical Analysis, to the today's program. Welcome you, sir.
Thank you.
It's my privilege to welcome the convener of our today's program, Dr. D. Ranganayakulu, sir, Principal and Professor of Sri Padmavati School of Pharmacy, to the today's program. Welcome you, sir.
Yeah, very good afternoon. Welcome you all.
Now, I would like to invite our honorable speaker, Mr. Nur Ahmad Syed Detta, sir, lecturer of Pharmaceutical Analysis and Quality Assurance in the Department of Pharmaceutical Chemistry, University of Gondar, Ethiopia, to this program. We heartily welcome you, sir, to this program.
Thank you, ma'am.
Yeah, welcome, sir.
I would also like to extend my hearty welcome to the Dr. Muhammad Rafi Mokla, sir, Associate Professor of Microbiology, University of Gondar, to this program. Welcome you, sir. With the best wishes of Chief Patron and Chairperson of Sri Padmavati Educational Institutions, Srimati P. Sulochana Madam, and Director of Sri Padmavati Educational Institutions, Patron of today's program, Sri P. Praneeth, sir, we will begin this program. So they conveyed the best wishes for this program. Let us begin this event with the invocation song.
Vakratunda Mahakaya Suryakoti Samaprabha Nirvignam Kuru Me Deva Sarvakaryeshu Sarvada.
Now, I would like to present a brief profile of our institution and the department to the virtual audience. I'm going to open the PowerPoint presentation. Within 1 minute, I'll start the profile.
Hello, Irshika.
Sir. Sir, I'm going to open the PPT. Is PPT visible?
No, it's not uploaded. It's not visible.
I deem it a great privilege to present the profile of our esteemed institution, Sri Padmavati School of Pharmacy, which was established in the year 1997 by late Dr. P. Mohan Rao Garu and persisted by the chairperson of Sri Padmavati Educational Institutions, Srimati P. Sulochana Ma'am, and director of SPEI, Srimati P. Praneeth Sir. The courses offered by the institute are Bachelor of Pharmacy, Master of Pharmacy, PharmD, and PharmD PB. And the postgraduate courses for the MPharm are Pharmacology, Pharmaceutics, Pharmaceutical Analysis, and Pharmacy Practice. The success of the institute for the past 23 years is emanated by the various awards and achievements of the institute. So various awards like Pratibha Awards in University Gold Medal and Campus Preparedness Award was received for the institute in the year 2015. We are proudly to say that we have more than 150 plus published journals at international and national level, and our students and faculty from the college has participated in various conferences by presenting oral and poster presentations. So in the institute, we have 8 PhD awarded faculty, and our students were qualified in every year competitive exams like GPAT and NIPER. 10 plus students qualify in every year GPAT and NIPER, and the college actively conducts the workshops and conferences every year. And these are the research scholars who are pursuing the postdoctoral degree at Sri Padmavati School of Pharmacy. So Sri Padmavati School of Pharmacy always encourages its faculty and provides all the facilities for the scholars to upgrade their qualifications. And this is about the best achievement of SPSP, the SPSP alumni who are settled in various positions. So SPSP alumni were settled in government positions like drug inspectors, research officers, central and state level pharmacists, in academics like university and college level professors, associate professors, and assistant professors. And the students, the alumni students were well settled in the industry as CEOs, managers, assistant managers, associates at various pharma industries, and they have started their own entrepreneurship like small and medium scale industries as well as pharmacies. And our alumni students are also working at the reputed hospitals and IT sectors as junior doctors, clinical pharmacists, and PV associates at various IT companies. So these are some of the activities, pics of the activities which are conducted at Sri Padmavati School of Pharmacy, like National Pharmacy Week celebrations will conduct every year, and will conduct all the competitions like debate, elocution, and essay writing, rally, NSS activities like rallies on the World Pharmacist Day. So every year we allow the students to have a glance at the industries, and we will conduct extracurricular activities. We have college clubs like Dance and Music Club, SAC Club, Credit Guidance Cell, Placement Cell, Grievance Cell, and Anti-Ragging Committee. And every year, annual day celebrations will be conducted at the college. And SPSP always stood first in the active participation of social welfare activities. And we also conduct alumni meet every year. And now coming to the Department of Pharmaceutical Analysis. So I'm happy to say that this year is the 10th anniversary for the Department of Pharmaceutical Analysis, which was established in the year 2010. So in the Department of Pharmaceutical Analysis, we have sophisticated instruments like HPLC, FTIR, and UV visible spectrophotometer. Various research works have been carried out in the department, like advanced works on QBD, chemometrics, and analytical method development. So we have experienced faculty in the department, and also the department contributes major publications to the institute. So I would like to show some of the pics of our alumni students, so who are working in various industries like Arabindo, and also Arabindo, Micro Labs, PRA Health Sciences, Germany, and various companies like Dr. Reddy's, as well as alumni who are settled in the colleges as assistant and associate professors. So these are the few pics of the Department of Pharmaceutical Analysis alumni. So thank you all of you. Now I would like to invite the convener of today's program, Sri Dr. D. Ranganayaklu sir, to present the opening remarks for this session.
Shika, is it audible?
Yes, sir. Yes, sir.
Hello?
Yes, sir. Is it audible?
Okay. That's indeed, it's my great pleasure to invite you all this the occasion. A very good afternoon to all. At the outset, I would like to convey my thanks on behalf of my management and my faculty and my students and the delegates and the students who are registered as part of this. I would like to convey my thanks to Mohammad Rafi Gatia, who was a resource person today for the program of vaccine development and all of the pharmaceutical sciences and the quality assurances. And at the same time, I would like to invite Dr. Mohammad Rafi Sir, who has joined. Of course, sir is one of the resource for tomorrow program, and I was very happy to invite him as a special inviter for the today program also. I invite on behalf of the management and staff for this present today program. As earlier, Preeti Shika has been narrated very much in the development of the department that is pharmaceutical analysis, M-Pharma. So almost it is completed 10 years of old, right? I mean, it is incepted in this 2010. Almost 10 years has been over. So many alumni has come. They have been reached at a good position, in the quality and the competence. But all credit goes to headed by Dr. Rajashekar and Dr. Sridhar. But Dr. Sridhar is one of the in charge for the both chemistry and analysis, and Dr. Rajashekar has been leader at present this pharmaceutical analysis. And this Dr. Charan Kirtishika is the main tools for this for the credential of the pharmaceutical analysis. Once again, I appreciate all the team, organizing committee for making this kind of program during the pandemic stage, because we have kind of serious program since June onwards during the pandemic stage, with the intention not to have the stopgap arrangement for updation of our knowledge and research skills in the pharmaceutical sciences. In view of this, we have promoted many programs. We have conducting this kind of web series. It has happened to be as an Indo-African web series for 3 days program. I especially invite today that speaker is Noor Ahmad here to deal this program, that is the preparation of vaccines, the role of pharmaceutical analysis, and quality assurance and the preparation. Definitely, so we have little exposure in the curriculum regarding this vaccination preparations and preparation and other conditions. We are lacking depth of this regarding the methodology of preparation. Definitely, this virtual talk is going to be enrich the wisdom of both experts, faculty, and research scholars. Definitely give a lot of impact on the research oriented and the vaccine preparations. So this again, once again, and thanks on behalf of my management and staff and students by welcoming for this program. I convey my thanks once again to Noor Ahmad here and Ahmad Rafi for this program. Thank you very much for organizing this hour. Thank you very much.
Thank you very much, sir, for always supporting and encouraging us to conduct this kind of webinars. Once again, thank you very much, sir, for your valuable address to today's program. Now, I would like to invite Kumari J. Bhavita, Assistant Professor of Department of Pharmaceutical Analysis, to present the profile of our today's speaker, Mr. Noor Ahmad Syed Getta, sir.
Shika. Yeah, just now I take the privilege and liberty. I'm inviting Professor Sridhar sir in this regard. Under the complete of program, I will take charge, and because I'm going to be there with some other AFC program, sir is going to be open operation. Please.
Yes, sir. Yes, sir. Thank you, sir. Thank you, sir.
Good afternoon to all the dignitaries. and our virtual audience. I'm honored to introduce our today's speaker, Mr. Noor Ahmed Syed Getaw, lecturer in Pharmaceutical Analysis at QA, former head, Department of Pharmaceutical Chemistry, University of Gondar, Gondar, Northwest, Ethiopia. Sir has completed B.Pharmacy in University of Gondar, Ethiopia, and has MSc in Pharmaceutical Analysis and QA from Addis Ababa University, Ethiopia. And Sir has more than 10 years of teaching experience in various positions like graduate assistant, assistant lecturer, and lecturer at University of Gondar, Ethiopia. And Sir is an active member of Ethiopian Pharmaceutical Association and Ethiopian Red Cross Society and many community service activities. Sir has also published many research and review papers in internationally reputed peer-reviewed journals and participated as co-investigator in many mega research projects. Sir has also received special training sessions from many universities in areas like GMP from Addis Ababa University and medicine dossier assessment from Ethiopian Food and Drug Administration and research ethics from University of Gondar. With this brief introduction, I would like to invite our today's session speaker, Mr. Noor Ahmed Syed Getaw, sir, to start our today's session.
Thank you, ma'am. Dear participants, academic personnel and scientists, I would like to say welcome, and I would like also to express how honored I am to be invited to deliver this speech at the Indo-African International Webinar Series as the first speaker. Thank you very much. So to begin my presentation, our today's presentation is going to be regarding vaccine development. We'll have a discussion on these topics, including what is meant by vaccine as an introductory remark, in vaccine development and testing stages, or what are the different actual stages that we should go through if we are going to develop and bring a vaccine to the market. And alongside that, we will also be dealing with quality assurance of vaccines, what quality assurance is, and why we should do this quality assurance will also be included. And quality control. Quality control is also included in this topic as part of quality assurance. And one significant step, one of the most critical things included in the vaccine development process. And finally, I would conclude my presentation, and that will be the end of my presentation. Okay, so vaccines are actually a class of biologicals or types of biological preparations or formulations which impart or which ignite the immune system of the host upon introduction. So different means. So what they actually do is they are jump-starting an immune response to a particular pathogen, or they will simply train our body to be familiar with an invading host before the actual host, the actual pathogen comes into our body. Or are doing all this without causing the actual disease if they are properly evaluated and if their quality is properly assured, they will not be causing the disease. They will simply make us familiar with this disease. So they will reduce the risk of actual infections by introducing our immune system and starting its work towards the fighting of the disease. So how are these vaccines doing this thing? It involves a series of things like recognizing the invading germ as a first step. It may be a virus or bacteria or any related pathogen. They will recognize that germ, and they will also make our body to make or produce antibodies, which are proteins prepared by the body as a mechanism of defending itself from the pathogens. And our body is set or designed to remember these things. And after these things happen, it will keep remembering the disease until a certain period of time, depending on the type of the vaccine. So that's how they make us defend ourselves from the diseases or infectious diseases. When it comes to the components of vaccines, there are four main or major components. The first and one of the most important thing is antigen, which can be regarded as the active pharmaceutical ingredient in any vaccine preparation. The type of this antigen may vary from live attenuated germs like bacteria or virus and other toxins or toxoids, or it may even be some part of the bacteria or some cellular component or any other biochemical substance can act as the antigen. So the other components are adjuvants or what we call excipients, which are necessary or intentionally added to the preparation to optimize the activity or the action of the actual antigen. And preservatives can also be used to maintain these vaccines throughout their lifetime or to keep its effectiveness for the desired period of time. Similar components known as stabilizers are also included in vaccines, which enable the protection of the vaccine upon transportation and storage conditions. So it will keep the vaccine to maintain its identity alongside with the preservatives. So most of or the majority of components of the vaccine may not be totally new things to our body, because these are mostly some natural compounds in the body or from some sort of animal, and they may even exist in the environment. In the foods we also they may be present. So they are more or less familiar things in vaccines, and alongside their ingredients are or should be highly checked and controlled in order to make them safe for usage, because they are actually obtained from some disease-causing entities. So the care and the precaution should be maximized in order to prevent such problem. And regarding the mode of administration, they are mostly prepared as injections, which can be injected using needles, or they can also come as oral vaccines, like the current polio vaccines, and they may also come as sprays, intranasal sprays for administration. Having said that, now I'll proceed to the different stages or the major stages of vaccine development. And while preparing for this speech, I have chosen to pursue the modality of the United States simply because that was where I could get information easily. And I will focus my discussion on the Food and Drug Administrations of the US procedures for the most part. So the vaccine development process is mostly lengthy and complex endeavor. So it's going to take more than a decade or so, something under normal circumstance, because it involves careful planning and execution of different major steps. So this process requires joint effort by the public and private organizations or different bodies or parties may involve in this development process. So different regions and countries of the world have their own regulatory bodies or drug and food administering entities. For example, when it comes to the United States, it's the Food and Drug Administration, which regulates vaccines and other pharmaceuticals alike. And they have this specially designated body in the FDA, specifically targeting vaccines regulation, known as the Center for Biologics Evaluation and Research. So it's through this department the FDA monitors vaccine development and production. When it comes to India, it's the Central Drug Standard Control Organization, as most of you might already be familiar. It's this organization that oversees the production, development, and marketing of pharmaceuticals, including vaccines. And the EU have their own regulatory agency, European Medicines Agency, and the like. And most other countries tend to follow the rules and regulations forwarded or recommendations forwarded from the World Health Organizations. So most of the countries may prefer to follow WHO's recommendations. So when it comes to the vaccine development, according to the Centers for Disease Control and Prevention of the US, there are these 6 major steps in the development of vaccines, including exploratory stage, preclinical stage, clinical trial or clinical development, and regulatory review and approval, and finally, manufacturing and quality control. So I will follow this pattern while discussing the vaccine development stage. And I would like to recommend you to constantly keep remembering the role of quality assurance and quality control in all of these stages, especially beginning and beyond the clinical studies. Starting from the clinical studies, things get more and more stronger, I mean regulatory aspects. So the first one is exploratory stage in the vaccine development. So at this stage, it's practically some basic level research conducted in different academic and scientific laboratories. It may be a governmental or private institution providing the establishments, the laboratories, and the like. So individual scientists may perform basic level researches to try to find some promising compounds as initial inputs for the pipeline or into the pipeline. So the search is mainly on virus-like particles or attenuated or weakened virus and bacteria or weakened bacterial toxins or toxoids and other substances derived from the pathogen. So what they do is they will simply check and try to see if there is any sign of immunogenicity or any vaccine-like behavior from this. And if there is something that is believed to be promising, then they will proceed to the preclinical stage of the development or the research. And at the initial exploratory stage, things may not even be that much coordinated and recognized. So everybody is doing everything they can in their own efforts. And the efforts tend to get more and more focused as it goes up the ladder of the vaccine development stage. And the second one is preclinical stage. So at this stage, there may be testing of that virus-like particle or toxoid or whatever it is being considered on animals like mice and monkeys or guinea pigs and the like. Primates, rodents and the like are used as lab test animals. And they may also use different cell cultures and or tissue cultures to check the safety and immunogenicity. Immunogenicity means the ability to ignite the immune response from the host or to start immune response by the host. So they will actually check this. And these preclinical studies actually will give us some hint regarding the cellular response which is expected from humans or the final targets of the vaccine. So what kind of response is going to be obtained is first obtained from this preclinical stage. And these studies may also give us safe starting dose for the next phase of research, which is the clinical stage. Which dose is going to be safe can also be obtained. Some level of information can also be obtained from these studies. And safe method of administering the vaccine, or how in the event of successful immunogenicity, how is it going to be delivered or administered to the intended host will also be obtained. But among the different activities done at the preclinical stage, adapting the vaccine, the candidate of the vaccine, or optimizing the vaccine to make it a little bit more effective is one of the activities. And the other one is doing challenge studies with animals. Challenge studies refers to the use of the vaccine on a subject or on a test animal, followed by the infecting of the animal with the actual pathogen. That is what we call challenge studies. And these may be done at this stage, at the preclinical stage. And for the most part, many of or almost majority of these candidates will not make it after this stage, or will not go to the clinical phase of the vaccine development. So they will lack the desired or the required immune response, and owing to that, they will just be left. But if there is anything promising, or if there is any sign of good immunogenicity or vaccine-like behavior, then anyone interested in funding that study will have to apply. For example, in the case of the US, they will have to apply to the FDA, the Food and Drug Administration, for an investigational new drug application. So this IND application is filed for products which have given some promise of being developed into a vaccine to be tested for clinically or on human subjects. So while getting this IND application to the US FDA, they will have to include a description of the manufacturing and testing processes, just how it is going to be manufactured and tested, and everything will have to be documented and submitted to the FDA. And summarizing the laboratory reports, the reports that have been done so far on the products will have to be summarized and delivered, and describing the proposed study, or what and how exactly are going to do regarding the clinical stage of development. So now coming to the clinical development, this is actual testing on humans, and it has got 3 different phases and an optional 4th phase. To begin with, phase 1 development, clinical development or clinical trial. This phase 1 development involves the use of a small group of humans, particularly less than 100, or at least a few dozens of healthy individuals will be used to check the vaccine candidates upon this. And things are not as tough as in phase 2 and 3 and beyond right now. So the goal of this phase 1 clinical trial study is to assess or to check the safety and to determine the type and extent of immune response in humans. Using actual human subjects, they will try to check safety and immunogenicity, extent and kind of the immune response. And sometimes the researchers may also follow challenge model. Like I said earlier, in the challenge model, they will choose to administer the actual pathogen, infecting the host, the subjects with actual disease-causing pathogen after the required length of time has already passed upon, after administering the vaccine. So in phase 2, these clinical studies involve more number of people or human subjects, typically by several hundreds. And these individual subjects will have similar profile or similar characteristics like age, physical health, and other conditions particularly similar to the population to which these vaccines are intended. So some simulated actual patient population, something like that, may also be included in this phase 2 clinical trials. The goals in this phase is to evaluate safety, immunogenicity, proposed dose, even schedule of immunizations, and method of delivery as well. So these things will be checked. And these trials are randomized and well controlled, and often they include placebo. Not always, but they will include placebo or some sort of solutions and related substance or formulations lacking the active pharmaceutical ingredient or the active antigen under consideration. Now going to phase 3. In this case, the vaccine product or the candidate is given to thousands of people and tested for efficacy and safety. It's all about efficacy and safety for the most part. And these are also randomized and double-blind. It's double-blind and randomized using several thousands of human subjects. But there is also the use of placebo, almost always in this case, unlike phase 2. So it involves this placebo. And the main goal will be to evaluate the vaccine safety in larger group in an effort to surface or to unveil some rare adverse events. So there are side effects, particularly which may happen in like 1 in 10 or 20,000 individuals. So in order to see the extent and the type of those adverse drug events, this is very instrumental. And when it comes to the efficacy check in phase 3, there are a variety of questions, answers to questions like, does the candidate vaccine actually prevent the disease? Using some challenge model, they will get answers to this question. And does it prevent infection with the pathogen? Is it capable of preventing actual infection? And does it lead to the production of antibodies or any other certain types of immune responses related to the pathogen? What is the actual mechanism of doing this or defending our body? All of these questions will have to be answered in this phase 3 clinical trial. And not always, but sometimes phase 4 clinical trials are also conducted by the manufacturers. So this is a lifetime responsibility as long as the vaccine is in the market, and that manufacturer will keep on researching the vaccine even after the market. So when it comes to the regulatory review and approval process, as I have previously mentioned, the regulatory review, the regulatory actions, quality assurance aspects are even included right at the moment of decision of trying or checking the vaccine on humans. Beginning from that point, the regulatory aspect gets tougher and tougher. So the FDA's specific wing for vaccine evaluation, or Center for Biologics Evaluation and Research, is responsible for regulating vaccines in the US. And the sponsor of these vaccine candidates should pass through these steps. All of these are regulatory hurdles. And the first one is an IND application or investigational new drug application, as I have already mentioned. In pre-licensure vaccine clinical trials, a permit to conduct vaccine clinical trials on humans. In the biologics license application, after the clinical trials, if it's still promising, then it's nearing licensing stage. So that's why they will apply to the biologics license application for biologics license application. An inspection of the manufacturing facilities to check the manufacturer's facilities using its inspectors. The national regulatory authority will have to perform thorough checks whether that facility is fit for producing something like vaccines. Will also have to be confirmed. In presentation of findings to the FDA's Vaccines and Related Biological Products Advisory Committee. So the findings or the results of this regulatory checks will have to be presented to this office, to this committee, and upon that receipt, they will base their decision. And usability testing of the product labeling, or the finished product, will also be checked. Regarding manufacturing and quality control, the FDA or the analogous responsible regulatory authority will have to check before allowing that vaccine manufacturer the marketing authorization. They will have to check the manufacturing or production sites and quality control facilities. Each and every one of these will have to be thoroughly checked. Coming to the quality assurance of vaccines, the term quality assurance is, as you all know, it's a vast concept. It's a management tool, and it includes anything and everything that directly or indirectly impacts the quality of a product from a manufacturer. So it's a huge concept, quality assurance. It can be viewed from these two viewpoints. From the manufacturer's perspective, quality assurance may be within the boundary of that pharmaceuticals manufacturing plant and immediately below the quality management. Then it will be in charge regarding everything or concerning everything about quality of the goods and products obtained from that organization. And from the regulatory authority or regulatory agency point of view, quality assurance can also be considered. So it's something they will check on the different manufacturers in organizations or pharmaceutical companies. Okay, and the National Regulatory Authority is responsible or in charge of making sure that every quality expectation is met, and this assuring the quality of pharmaceutical products gets particularly difficult or tough in the case of vaccines, owing to their biological nature. These vaccines are biological, and the vaccines themselves may be source of some huge catastrophe. So to prevent this, the quality assurance process is very difficult in the case of vaccines. Certain disastrous event may even happen if the quality control and the quality assurance process is a bit relaxed. And I think most of you are familiar with the Qatar incident of the United States, which happened in 1955 on a polio virus vaccine. The polio vaccine, they thought they had done everything the right way, and the deactivating or attenuating of the polio virus and everything, but that was not the case, and disaster struck within a few days of the vaccination of about 100,000 children. Okay, so for this and other related reasons, quality assurance is an essential concept to apply. And the regulatory authority has the responsibility to confirm that manufacturers adhere to approved standards like GMP and other related norms, like national and other requirements for manufacturing, as well as quality control of pharmaceutical products. So it's really an essential component. And the mechanism or how they are going to effect this may depend on these two things: the availability of resources, what types of resource facility are available to do this quality assurance work, and whether the product is local or imported. Things may not be the same for some local product and something imported from abroad. So this will have an effect on the decision. And biological products are different from other ordinary pharmaceuticals due to their biological origin or from their source, and they are also complex in nature. Their molecular structure tends to be more complex as they are obtained from biomolecules. and they may require special considerations, special quality considerations to maintain the goodness of these products. Like the starting material itself is probably biological in nature, the manufacturing process or how they manufacture it may even be dependent on some sort of life form. The test methods needed to characterize these manufacturing batches may also themselves be biological in nature. So there is a huge concern in handling of these vaccines and other biologics or biological preparations. Coming to the quality control of vaccines, so quality control is one component of quality assurance under GMP or good manufacturing practice, and it's concerned with the testing and the conduct of actual laboratory work or taking samples and checking their qualities and documenting what is being done and the like. So the vaccine quality control, it tends to be a little bit more stringent and difficult. Vaccines may be one or more of these things, live attenuated pathogens or germs, inactivated or killed antigens, toxoids or inactivated toxins, and even subunit vaccines. So regarding the quality control methods available for vaccines, things are a little bit difficult because even more difficult than other biological products like hormones and therapeutic molecules or antibodies, because vaccines are heterogeneous. When you say vaccine, you cannot tell whether it's a living thing or it's just some component of that germ or some sort of biomolecule. So there are different types of vaccines, and that tends to make things difficult for the quality control department. But in the case of other biologics like hormones and therapeutic antibodies, they can be checked, or the quality of these products can be checked using more or less straightforward techniques like electrophoresis, nephelometry, MS or mass spectrometry, and the like. We can also use these same techniques on vaccines, but in the case of vaccines, due to that heterogenic nature, things may not be as easy as the other products. It may not even be capable of telling the actual quality status of that vaccine. So this is the problem. And the presence of high quality assays or tests in the development stage of a vaccine can reduce huge risk to humans and in the clinical trials. So we need some high quality assays or tests. By high quality, I may refer to the exactness or validity of the data, or its meaningful data will have to be presented. And these assays required to show or to demonstrate this quality may depend to a huge extent on the type of the vaccine. The type of vaccine under consideration decides which assays to use. But we may not need to use the same tests for live attenuated vaccines, or in the case of some sort of just a biomolecule, a protein or some other type of biomolecule. So the specific techniques or methods required may vary depending on the type of the vaccine. In post-licensing, this number of tests or the required number of tests to be performed in the vaccine may get smaller, reduced in number. Because in order to be licensed, that manufacturer must have demonstrated some efficiency in the production process, and in the production process, they have shown enough level of consistency, validated methods of production, quality control, and the like. Owing to these reasons, the actual number of quality control tests tends to be smaller post-licensing than during the licensing or the vaccine development stage or phase. So some certain selected test methods can be used to assure or ensure the quality, potency, and safety of the vaccine. And regarding these techniques or methods, specific quality control procedures, there are two main categories of techniques of vaccine quality control. Or we can mainly divide the huge number of tests available to be done on vaccine products into these two groups. The first one refers to the techniques for measuring biological activity or active concentration or active conformation of the vaccine, meaning the antigen will not be directly quantified or identified. So the presence and the type of antigenic substance or the active pharmaceutical ingredient in the vaccine will be indirectly assessed using its biological response or activity in the host. These are also known as potency assays or potency tests. And in this class of techniques, the second one, which we will be discussing ahead, is those techniques used to detect and quantify the antigen directly. So these first techniques or potency assays, they include animal tests, cell-based assays, and different physicochemical and immunochemical methods are included under these divisions of methods or quality control tests, and based on the measurement of one or more parameters which are believed to be linked directly or indirectly with the efficacy of the vaccine under consideration. So these techniques will not directly quantify or detect this. The aim is to measure something as a result of the effect of that vaccine in the host's immune system or in the host animal's body. So the most common potency test in this case is based on the immunization challenge procedure. It's still common until this date. The most majority of these methods is based on the challenge procedure. So these models in this challenge procedure, first they will expose the test animal. They will treat the test animal with a vaccine, and then they will check whether the vaccine is working or not by actually trying to infect it with the pathogen itself. And there are certain limitations to this challenge model, and these limitations are related with the animals used. When we choose to use animals, it has its own complications owing to different reasons. And the method is by itself expensive and time consuming. The expensiveness is also related with the animals, animal handling and everything related to it. And it's also time consuming. It may take months, like 2 months, to get the results back, the results of a vaccine evaluation, which is very difficult, especially considering the short life of these vaccines. Vaccines, most of the time, they're going to be useful for 2 years or something like that. In that 2-year length is going to be compromised due to these animal-based challenge tests. So in these potency tests, there are 4 classes of methods. The challenge test, serological analysis. In serology, it's almost the same as the challenge test, but they will not be actually injecting or infecting the animals with actual pathogens or disease-causing entities. What they do is, after administering the vaccine to the animal, the test animals, they will keep for the required length of time, and they will try to see some markers, something from the body, like antibody or toxin or something like that. And they will try to quantify that. In that target of analysis is something as a result of exposure of the animal to the vaccine, but they will not be challenging the animal with the pathogen. In the third class of these potency assays is cell-based assays. So these assays are based on cells as their name clearly states, they are cell-based, and they may not make use of these animals in everything. And titration assays can also be used for potency assays. So if we choose to use in vitro techniques or in vitro methods, that's going to enable us not to choose animals, or we can avoid in vivo models, and we can also together avoid the complications or problems of the in vivo tests. In addition to this, the animal models may have several different shortcomings or limitations, like will they will be relevant or not? That's one question to be answered. And reliability itself, the response that we may get from an animal varies from animal to animal. It's inherent in any living entity. One animal may not be exactly the same as the other. So that's another challenge. Cost is one limitation, and moral acceptability in animal handling, especially in those tests involving actual infecting of the animal with the pathogen or the challenge model. In the case of the challenge model, the animal's right and handling will be hugely compromised, and the animal is evidently going to suffer a lot. Coming to the techniques for measuring the concentration and integrity of antigens, these techniques are used to directly identify and quantify the antigenic entity in the vaccine or the active pharmaceutical ingredient. In this case, we have different classes of techniques like spectroscopic or colorimetric assays, and different separation techniques like chromatographic, electrophoretic, and related techniques are available under this class. In their use of these colorimetric and analytical separation techniques are for the quantification of intermediate and final product or the active pharmaceutical ingredient in the vaccine during manufacturing process or as in-process quality control measure, and at the end of the manufacturing process or in the final product. And they are also used for detecting impurities or unwanted contaminants in the vaccine. So, these techniques can also be used. So colorimetric or spectroscopic techniques are generally easy to perform. That's one big advantage. And they can measure, among others, these features of vaccines: protein concentration, polysaccharide content, number of primary amine groups, and cell DNA can be effectively determined using these techniques. By the techniques, I mean colorimetric and separation techniques. So, separation techniques may include chromatography and electrophoresis, not just gel electrophoresis. Capillary electrophoresis has also shown impressive results. Liquid and gas chromatography, even gas chromatography, is also possible to be used. And chromatography is very instrumental in this one. And they are often used to determine the stability and purity of protein and polysaccharide vaccines as components from the pathogen. These things will be directly quantified and identified. The other method available is mass spectrometry. So it's currently its use is seeing some astonishing rise, and it's used to characterize antigens and impurities. And identification, fragmentation, and other structural degradations, which are peculiar to mass spectrometry, are the applications. Using MS, we can see things different from other analytical techniques. So this is one huge achievement. There are also other various techniques available, even from spectroscopy, those which I did not mention, like infrared spectroscopy to characterize and to optimize the identity of the vaccine entity. Others like fluorescence-based spectroscopic techniques analysis or fluorimetry, even polarimetry, circular dichroism and the like can be used. UV-Vis spectroscopy is also commonly used. All of these can be very instrumental. And beyond this, also immunochemical techniques like ELISA and the like are also available, but we will not be having detailed discussion in none of these. And to conclude my presentation, the development and evaluation or licensing of vaccine product is a lengthy and challenging process, may take more than 10 years, and it also involves several preclinical and clinical stages that need to be carefully planned and executed. And along this, quality assurance is essential and is aimed at ensuring the safety, quality, and efficacy of the final product. So these are the references which I used in preparing for this speech. And thank you very much for your time. Thank you.
Thank you very much, sir, for the wonderful and informative lecture regarding the vaccine development and highlighting the stages of vaccine development, importance of the role of quality assurance, as well as what are the analytical techniques which are used in determining this concentration of antigen. Really, it is a very useful session, sir. Once again, thank you very much for providing this kind of topic. Now, so let's move on to the question session, sir. Some people are having doubts. Sir, a question is, what are the best sample preparation techniques used for biological compounds?
Okay, should I keep answering these questions or?
Yes. Yes, sir.
Okay, thanks, ma'am. So the sample preparation techniques used for biological compounds, they differ from substance to substance, from method to method. But in all of these sample preparation techniques, we need to be very careful since we are dealing with some sort of life form. We should not treat the sample preparation of these biological compounds or vaccines as in the same way as other ordinary pharmaceuticals, because that life form may be inactivated or it may be damaged during the handling process. So we need to be very careful. Otherwise, there are different sample preparation schemes, and we cannot deal with them in such short time. So what we have to know is that sample preparation needs to be very careful since we are dealing with life form.
Yes, sir. Thank you, sir. So the next question is, what are the guidelines applied for vaccines testing apart from the method validation guidelines which are prescribed in ICH? Is there any other guidelines, sir, applied for vaccine testing?
All I know is some country-specific regulations, like those developed by the FDA, and for regions like in the European Union by the European Medicines Agency. In countries having their own pharmacopoeias, they have their own specific guidelines. Some are harmonized, as in the case of the ICH guidelines. So there are also WHO recommendations or WHO guidelines. So there are these options. There are several options.
So next, what is the most effective analytical technique which is used for the analysis of vaccines efficiency? Most used.
Oh, most efficient. This is somehow, difficult. Meaning we are dealing some life form, as I have mentioned repeatedly. The most efficient, I will have to say, will be the ones utilizing animals, using lab animals, because if we rely entirely on some sort of physicochemical technique, it's going to be difficult to make sure that the vaccine will actually work inside the living host or human beings. That's what makes actually the vaccine evaluation very difficult. The mere presence of the antigen in the final product may not guarantee an immune response in the host. So, as to me, or according to my opinion, the best method would have to be something animal-based. If it's capable of eliciting immune response in the animal, then we can tell that it is going to do the same thing to humans. I prefer those.
Sir, one more question, sir. Although vaccines are prepared as per guidelines, how to minimize the dermal shock in clinical practice? Sir? The ways to minimize.
Thermal shock.
Yes, sir.
Okay, meaning pyrogenicity. Just if I did not misunderstand this question, I think it has something to do with pyrogenicity. Pyrogenicity means if the vaccine has something that can cause such febrile situations in the patients, then that is going to be dealt with using pyrogen testing. There are different methods available to check the presence or absence of a pyrogen in any vaccine product. It may be an in vitro test like the LAL test, or it may be something animal-based using rabbits and related animals. So, both of these techniques have their own pros and cons, but there are methods for checking whether it's going to cause this thermal shock or not in the actual subject. So it would be by checking pyrogenicity using pyrogen testing techniques.
Sir, one more question from my side, sir.
Okay.
Sir, are there any vaccines being under consideration in your country, Ethiopia, sir, currently for COVID-19?
Not scientifically confirmed actually, but there was some traditional medicine-based process undergoing in the Ethiopian Public Health Institution Pharmacognosy Research Lab. But I highly doubt it's going to be a vaccine product. It's not a vaccine product under consideration. But there is some sort of product, some possible candidate for as a treatment, not a vaccine.
No. Thank you.
Thank you very much, sir. Now, I would like to request the Organizing Secretary, Head of the Department of Pharmaceutical Analysis, Dr. K.K. Rashika, sir, to present the closing remarks of today's session.
Thank you, Kirtisika. So, distinguished webinar panel members, participants, ladies and gentlemen, good morning, good afternoon, and good evening to all. Today's session is the first part regarding the different aspects of the Indo-African International Webinar Series, which demonstrates the challenges of vaccine production, development, and drug discovery. However, this is the first held during the COVID era, which increases its importance in the face of new demands. I would like to underline few points in the reflections that I felt today. Despite the enormous progress made in the last 3 centuries, since Edward Jenner demonstrated immunity to the smallpox, the COVID-19 pandemic has exposed the critical imbalances in our medical system. Undoubtedly, top priority in pharmaceutical industry is to search for an effective vaccine, hopefully in 2021. In the meantime, the whole world is wishing for efficient drugs that would act against coronavirus. Pharmaceutical analysis, among other topics in bioanalysis, has been playing a vital role in the recent research needs. Already existing and new pharmaceuticals were examined with regard to their potential efficacy and activity against COVID-19. Therefore, the The analytical scientists are asked to be ready to perform at their best to develop and validate methods for all drugs under investigation. Furthermore, it was clear today, as highlighted by our esteemed speaker, Mr. Noor Ahmad Syed Geta, how vital the contribution of analytical chemists are in vaccine development processes. From our speaker's perspective, with his knowledge of excellence to the pharmaceutical analysis and quality assurance, even the pre-existing analytical methods must be re-examined, optimized, and revalidated. Speaker also stressed the importance of analyzing biological fluids, especially the blood serum of the drug administration requires sample preparation using modern pre-treatment techniques to be compatible with the modern sophisticated analytical instrumentation. So it's been a pleasure being with all of you today. So all of you work smart, stay safe. Thank you and good day. Go to Kirtisika.
Thank you very much, sir. Now, I would like to request co-convener of today's program, Head of the Department of Pharmaceutical Chemistry, Dr. C. Sridhar sir, to present the certificate to our honorable speaker, Mr. Noor Ahmad Syed Geta sir. Certificate presentation to
A warm good afternoon to all the participants and the dignitaries of today's webinar. It's my privilege and honor to present a token of appreciation to Noor Ahmad Syed Getawe for his valuable contribution on the day one of this Indo-African International Webinar Series. Please, Noor Ahmad Syed Getawe, please accept this certificate of appreciation from the organizing committee of this Indo-American International Webinar Series. We thank you for your immense contribution and sharing your experience of development of vaccines and the role of quality assurance and quality control in the production of the vaccine. So once again, thank you very much on behalf of.
Thank you very much, sir.
Over to Kirti.
Thank you very much, sir. Now, I'd like to request coordinator of today's Indo-African International Webinar Series, Dr. A. Srinivasacharan sir, to present the vote of thanks to today's session.
Thank you, Kirtishika. I am Dr. Charan, coordinator of today's Indo-African International Webinar Series 1, Part 1, presented by Mr. Noor Ahmad Syed Getaw from Ethiopia. And it's my privilege to propose the vote of thanks for this program on the first day of this International Webinar Series. First of all, I would like to convey our sincere thanks to our respected speaker, Mr. Noor Ahmad Syed Getaw. Mr. Getaw, thank you very much. I would also like to convey my heartfelt gratitude to our Chief Patron and Chairperson of Sri Padmavati Education Institution, Srimati P. Sulochana Madam Garu, and Patron of this program and Director of Sri Padmavati Education Institutions, Sri P. Pranit Garu, for providing all the support and encouraging us to do this kind of sessions. Thank you very much, sir and madam. Next, I would like to extend my sincere thanks to the Principal of Sri Padmavati School of Pharmacy, Dr. D. Ranganayakulu Garu, and co-convener of this session, Dr. C. Sridhar sir, for their continuous motivation and support for providing us to conduct this kind of webinar, online webinar successfully. Thank you very much, sir. A heartful thanks to the organizing secretary, Dr. K.K. Rashekar sir, for making this webinar happen, for contacting all the international speakers and making this webinar, international Indo-African International Webinar Series happen. Thank you very much, sir. This would be one of the wonderful achievements by our department. Thank you very much, sir. And I also extend my thanks to all the faculty of Department of Pharmaceutical Analysis who have contributed for the successful conduction of this part 1 of International Webinar Series 1, and all the other colleagues and webinar committee members of Sri Padmavati School of Pharmacy. But not the least, I would also like to thank all the participants, delegates who have attended and registered for this international webinar series and have been uh, watching this webinar live through various social media platforms like YouTube, Facebook, and Twitter. Thank you very much for all your active participation. Thank you all for giving me this opportunity. I also thank Dr. Mohammed Rafi sir for patiently attending today's program. Uh, Sir, we would like to meet you tomorrow for your part 2 program on vaccine, sir. Looking forward to meet you tomorrow, Dr. Mohammed Rafi sir. Thank you very much.
Once again, thank you very much to all the organizing committee members as well as students for making this webinar series part 1 a grand success. So hope this session is useful to all of you. Tomorrow we'll continue in the part 2 series by the speaker, Dr. Mohammed Rafi Mokla sir. Let us end this session with the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे